the stage.
Tom and Aldevron. It's been a really great company and people to work. It's really nice to go out there and not worry about the constraints of the industry or technology and work together. So thank you, and I think all the partners here really appreciate the work you're doing. Michael, thank you for the platform. Talk to you today. This morning was quite enlightening. You heard a focus around manufacturing. Towards the end of the science part of the talk, we're focused on that manufacturing part, not only just for research, but also once we get to cell and gene therapies. Still very early days, and so I'm not going to talk too much about that. It was a nice boost to the financing that we got. But what we've really looked at is building platforms. So I'm going to change gears here. There's a lot of molecular biology, a lot of protein biochemistry going on today, and that's awesome. We're focusing on engineering, okay, and building platforms. And there's a real drive. It really does translate itself to cell and gene therapy. And that's around scale. And how do you drive scale in a way of biology that's very complex and difficult to get consistency around publications and around different platforms and applications? So we've been focused on that. And so I'll tell you a little bit about our story. Tom asked me to talk about this story. It's unique. As Tom said, I've been with Bio-Rad Laboratories for a long time. It's a traditional life science tool developer. Scale is awesome. But when you need innovation, it's very difficult to do. You have to think differently. And so we focus on that. But you have to know what the problems are in the industry to address, to solve for these issues. So the macro challenges in biology and industry itself is that R&D returns, the return on investment in R&D is down, and quite a bit, and it's much more expensive. And if you start to see some of the press around the costs of therapies, they're going up tremendously. In fact, they're very high, and that limits accessibility. And I think Matt, you saw this morning around him talking about Africa or South America and trying to bring gene therapies there. There's no possible chance to do that right in an economic. Costs about $2 billion to bring a drug to market. That's gone up. My 20 years in the industry, that number used to be like $500 million or $500 million, now $2 billion. Incredibly expensive, lots of capital commitment and risk. And finally, what we're seeing in any emerging technology area that we've seen, I've lived through the PCR world, the NGS world, in the early days, trying to find standards and reproducible results is difficult. Complex that with the number of cell lines and different techniques we've talked about just in this morning, it makes it difficult to find reproducible techniques. So it is early days in genome engineering. Standards. What are the bioinformatics standards? I'll show you some data around looking at outcomes and the variability of the outcomes. We don't have standards yet around the bioinformatics. We're still learning. The tools are better, but they're not good enough. And then editing protocols. What are the manufacturing tools and ways that we're going to look at to standardize around? I talked a bit about access. Can we give people, there are a lot of researchers, a lot of students in the room, do you have access to the best tools out there to give you 80-90% editing efficiencies or guaranteed functional knockouts of your experiments? That's what you should be demanding. That's what you should be getting. Okay? Scaling is expensive. Doing an experiment a couple times on the bench is not too difficult. But if you want to do hundreds and hundreds of experiments, the capital cost is very high, the expertise you need, and it limits it just to a select few. And again, the applications are numerous. If you look at them, in vivo, ex vivo, gene therapies, these don't even include the ag applications. Numerous cells, numerous delivery challenges, numerous off-target challenges we just talked about. This is quite a hill to climb. Well, let me tell you a little about Synthego. Synthego, we were founded 5 years ago, commercially came on the market 2 years ago. We're founded by Paul and Mike Dabrowski, the refugees, as I call it, from the SpaceX program and Elon Musk, working for him in the 2000s. But what they did is they cut their teeth on how it is to work in a startup. I'm going to talk a little bit about the platform and philosophy we do to approach the challenges going forward. but you need help. So we just talked about capital help through our venture capital partners, but also some leading advisors. So Jennifer Doudna's on our advisory committee, along with Matt, who just joined. And also a key member, founding member, was Sir Andrew Whitty, who's the ex-CEO of GSK. It really helped us put the point on where we need to go around therapies. And then finally, as a startup, you got to be disruptive, right? That's the code. And so we took a look at building a platform around guide RNA, making single guide RNAs. 3 years ago, there was not an economic option out there. People were spending thousands of dollars to get guides to do a couple experiments, and that just wasn't viable. We saw we can make an impact there. Recently, we've launched a engineered cell line, as I'll talk about, a cell program that we're also feeling is quite disruptive and brings access to researchers. Okay, so let's peel back the onion a little bit. We think about biology as a data science. Yes, we make cells, we edit, but ultimately we're looking at data. And that data informs us of what next to do. Now I'm going to take the engineering approach. The engineers hate this top part of this slide, right? A lab. We're all very comfortable with a complex, convoluted lab where we have a lot of people pushing things around. Engineers, especially in the tech industry in the Bay Area, they see things in the bottom, okay? They see server farms, massively parallel automated systems that are derived into its basic units. And then you throw as much as you can at them, work them hard, and you get data out of it. It's an interesting approach. I'm a scientist by training. I feel like I'm an engineer by adoption, okay? So this is what I would call the secret sauce, okay, of what we're doing. We're focused on genome engineering. There's nothing mystical here, but it is a system that you have to practice and discipline. Focus on genome engineering, design, build, test, learn, right? And that learn is really critical, and there's great tools out there now around learning. Put that around engineering aspects of you build rapid iteration. We talk about that as agile software. We do that in hardware. We do that in every aspect, marketing. We actually do that in product development. It's rapid iteration on everything. An insane learning process. We build for automation. We don't build manual, we go for automation. We may not institute all automation at the front end, but we automate. Parallelism, massively parallel. The server farm, that's the way we think. We can synthesize thousands and thousands of guides in a day. And then scalability. Being able to replicate that, not only in the factory, but be able to relocate that factory in different places. And you can see that has a lot of application to what we heard about this morning. But you can't just do it with engineering principles. You need also great mechanical, electrical engineers. You need bioinformaticists, chemists, biologists, everything to do that. And the way I say is we put it into a crucible. We're a very small company, work very long hours, and you heat that up. When you heat up that, you get outcomes that rapidly get outcomes that you can leverage. And this is very, very critical for us as we go forward. So this is Tom asked me, this is the thing that feels a little bit different, because we have this cross-functional debate all the time, every day, probably every hour, about what we should do and where we should go. So it's quite exciting for me to be in this environment. So let me make this more tangible. So let's do a CRISPR edit. We design our guides, obtain our Cas9. We all know where to get that. We need to optimize the transfection methodology, edit, analyze. Then if we're going for clones, go into clonal expansion, and then finally check for clonality. Great big buckets, have it, but what happens is that there's 75 discrete unit actions in this area. Now, some of those are just ordering and procurement opportunities, but most of them have to go down to its base level that you need to take a look at and then build back up in the workflow. This is very critical, all the way from, again, designing and obtaining reagents to doing the edit. There's quite a few things around doing edit. I'll show some data around optimization. And then moving to clonality, making sure that your clones actually carry the edit through to the end is quite critical. So this is a platform that we've built out, and it delivers great results. This is a 384 transfection in an RMP format into HEK and 293 cells, pretty robust cells, good, nice trainer cells, cell line. And we see very reproducible results across the plate, quadrants, so we can subdivide it for scale. Quite nice. But one of the things that's real important is around optimization. And this is where the accessibility to scale helps. So every cell line we look at, or we operate with, whether customer cell line or our own cell line, we optimize. We use over 200 conditions. We run this over a couple days, and you can see that the recommended protocol from the published protocol gives only about 7% editing efficiency. And then only about half of the conditions give almost no editing efficiency. But if you look at different concentrations of Cas versus the guide, different transfection conditions per line, you can optimize this, and we've done this for every cell line. And now you can take a look at and get very reproducible editing efficiencies across multiple cell lines. We have immortalized cell lines, we have iPSC cell lines in here, T cells. You can get these very, very high 90+% editing efficiencies. We've actually expanded that out in our manufacturing automation platform to over 250 cell lines in optimization. Not all of them work out, and we're moving our way up to the 700 cell lines. So quite impressive in terms of scale and flexibility on the platform. That's an example of taking a look at engineered cells workflow. Behind this is our first platform we built 2 years ago out is automating single guide RNA. Basically, 2 years ago, you buy a 2-piece system, CRISPR tracer, and nail them together and use it. The protocols then were not that great. They're better now for the 2-piece, but really the solution to this, especially if you're starting to go to the clinic, is around providing single guide RNA. And so we developed a factory to do that. And the issue here is around control and closing the system. Oligonucleotide synthesis really didn't have a closed system, off-the-shelf closed system. So we built our own synthesizers in the vein of what you saw with the server farm, massively parallel. Took microfluidics, chemistry, biology, automation, and then wrapped that with a LIMS system that we track everything. We're insane about how we track all the data, okay? Because we want to make sure that everything is well controlled. Some outputs last year, we synthesized over 250,000 guide RNAs. This year it's much more. We've now played them out in individual guides by gene in a well or in library formatting. Talk about that in a little bit. From microgram to gram scales, we will be bringing us a GMP version of the sgRNA this next year. So it's quite exciting. Lots of inbound interest on that. We're working with NIST around standardization. Again, trying to get standards is critical if you're going to build an automation platform. And we want to support the bench to clinic transition, and we're doing that now. Okay, so talked a little bit about the factory that we've just built and for sgRNA. That's for DIY work. If you want to do your own editing, we will do editing, or we just released our engineered cell line, which is based on our second factory. I laid out a bit of the roadmap there. But what we're looking to do is make genome engineering a product. Make it simple. Okay? This isn't for the advanced research. This is for most researchers who just need cells. You need cells to do the experiment, then why do you need to go learn CRISPR unless you're doing CRISPR research or you're improving the process? A lot of people can do their basic experiments with knocked out cell pools. They're at least 50% edited and can deliver a functional knockout. And you can get those in about 4 weeks. You can get clones around 8 weeks. And so all you need to do here is put in the gene in the cell line. In 4 to 6 weeks, it will come 2 million cells will show up at your door along with the report. We do have ability to do iPSCs and T cells as our advanced cells, but the whole goal is to take those, which we're still training our systems on, and bring those into a product standpoint. Again, it's to get you to your next experiment as quick as possible and drive standards. We use consistent processing throughout we use a consistent process throughout the whole workflow so that you can, from order to order, you'll get the same results. And many of you have used in the room or have published with our guides. We've been very excited about the results that are coming through and the quality of those. Let me shift gears here that was around platform development. Looking a bit about the bioinformatics. Without good bioinformatics, obviously, your experiments are not going to work well, and we need better data to generate better models to then get better outcomes. So we've developed two tools. One's a very common design tool, good heuristics on that. There's many different tools out there around design. But what we're really excited about is the analysis side. It's taking a look at how do we analyze the edit. And so we released, in January, we released a tool called ICE. What it is all about the repair, looking at the repair from Sanger sequencing and looking at the of the repair. And we use visualization up on the top, the right side that you're looking at. We give you a visualization of this, the detail around the edits and the knockouts. And so it's quite easy to determine what's happening. This is very quick analysis. It can work up to 200 base pair deletions. And we've also shown that on the bottom right, good correlation to NGS. So it's very fast, and it lowers the cost of doing analysis, which we really like. Oops. Right. So being able to do batch mode is really critical. People are doing many edits. And so we expanded the system to be able to handle up to 700 sequences. And this is an example of 500. And on the right side, you'll see a KO score. And a knockout score, there's editing efficiency, but that always doesn't lead to a functional knockout. If you're in a multiple of 3, or you may not see a knockout. So the way we score that is anything that's not a multiple of 3 or a 20 base pair knockout. Anything bigger than that's a knockout score. So it really gives you a good idea whether you're going to have a knockout or not, functional knockout. Oh, by the way, those 500 sequences only took 4 minutes to do the analysis on, so you get rapid feedback. We're in open beta right now on our website around a knock-in tool. Allows you to get a knock-in score and evaluate that. So this is 190. Sorry, I can't see it, but 190 base pair knock-in, quite good size and efficiency. So we are big believers in multiplexing, so designing for 3 guides, and so you can actually get a dropout of the sequence if you're focused on knockouts. And so we've built the tool to handle multiplex, so you can put the 3 guides in there and see what the knockout. So this is the larger knockout range, and this is very powerful. We've brought the multiplex into our library sequences and strategies, and also our engineered cell strategies. I'll skip that. So multiplex knockout, why look at knockout versus just editing efficiency? If you look at the 60 genes that we knocked out with a single guide, they all had relatively high, not all, but most of them had relatively high editing efficiency. And if you use multiplex, you can see we can really gain improved knockout or editing efficiencies. But when you go and look at the knockout score, you really didn't see that good of knockout on the single guides, but multiplex is much higher. So we're very excited about people using this score and talking about knockout instead of just editing efficiency. Because ultimately, you want that knockout to lead to a functional knockout. Again, we've multiplexed out our libraries. They're all here. These are available if you're looking at preformed libraries and then a whole genome library. One of the things we found is comparing with our RMP format. versus lentipool formats, which were basically used to generate the data sets and design tools that are listed up on the chart, is that there's not a correlation from when we run thousands of guide RNAs versus what we see coming out of these tools. And that's we find very interesting. So it really depends on the way you generate your data set and train. And we've trained over thousands of guides into our data set. So we want to build better models and be predictive about this. And so we've looked at in silico prediction tools. And if you look at the chart on the left, the indel distributions, you can see that the predicted versus the observed indel distribution is quite predictable. And what this allows us to do is actually pick better guides and take the information back into giving better guides and prediction tools. So we will not need as many guides to be able to get a functional knockout. Okay, I'm running a bit up against the time clock. I'm also right before lunch, so I know you guys want to eat at some point. Synthego's focused on a couple things. It's on standardizing genome engineering and providing access to people who don't have the best tools. Focus on scale and accessibility for genome engineering in terms of generating a lot of data. And actually, we have a good viewpoint towards cell and gene therapies and how we would like to do help with the manufacturing processes and challenges there. So I'd like to thank you for your time, and we will take any questions.
Thanks, Ted. That was a great talk. Yeah, the idea of solving complex biology problems with engineers is something that struck me when I met with the team out at Synthego. I was interested on your slide with flow chart where you had like the big block, and then you had the flow chart, all the flow charts and all the unit operations. I'm envisioning like a conference room with sticky notes all over the side of the wall mapping out those flow charts. But my question is around kind of, do you have like specific like manufacturing lean techniques that you use to continually iterate, those processes from an efficiency standpoint? I get it's in the ethos of the company, but there's probably more to it than just that.
Thanks, Tom. So the way we want to do it is we want to censor everything and take feedback. And so every system we put in place, we have a feedback loop that there's a machine learning component of it that brings back that process. So there was a room that was all sticky notes, but really it came down to the point of getting the right people in the room and hammering out that unit operation, maybe consulting outside people for some guidance since we may not have the expertise in the room. And then making sure that we measure at each step, quality step along the way. And we do this in a way that is sense and it's always available. So we have visual boards that are up in the manufacturing site. You can see where things are, where they're failing, what synthesizer's on, how it's going. Everything is mapped out. I can do it from home or anywhere. So really exciting. This model approach is applicable to any part of that workflow. Just part of our.
Any questions? Maybe one more question, and then we can head out to lunch, and you can obviously talk to Ted at the breaks, and he's here tomorrow also. Can you tell me when someone orders one of these knockout cell lines, what do they all get with the package? I know we saw a little bit about the data that you showed, but what could someone expect? And I like the idea of, if you're not great at CRISPR, why set up the whole lab to do it? And just what you want is the end game is the knocked out or the edited cells. So maybe you can explain a little bit about that.
Sure. So what you get in the knockout cells, you get 2 million cells of edited cell lines plus the ICE report, which shows you the percent indel and frequent distribution. You get the wild type cells, 2 million, so you can do your comparative studies. And basically that's all you need right there, and then you can go on to your assay, your next step. We do recommend primer sequences. We can provide those for you from a sequencing or PCR. We want to make it as easy as possible on the pack. There's too much, we fail a lot on the sequencing side or the primer side in getting those sequences correct. And sometimes I have to resequence, and we find that quite frustrating. So we built in-house tools to make those better, and we make those sequences available to researchers when they purchase around the genes.